Many of you will have had the problem that you turn on the light and nothing happens. Now, what could have gone wrong? There aren't all that many possibilities. So it's either the light bulb has burnt out or there is a power outage. And I can imagine that you will soon come up with an experiment how to separate between the two. Biologists, molecular biologists, have the same problem very often in their work. So imagine that some external stimulus comes to a cell receptor, which then passes on this information to turn on a response gene through an activator and inactivator. And you might have two different models for this system on the left-hand side and on the right panel. During my PhD project, we developed methods how to design optimal experiments so that you can tell the difference between these two possibilities with your experiment. So the way I see this is that there are many experimental biologists working on particular problems, and they try to dig deeper and deeper. Over the past decade or two, their work has been extended by the help of systems biologists, who essentially model biochemical systems in computers to help the experimentalists interpret their data. And where I come in is that I'm in a third layer trying to cut across all these fields by identifying commonalities and developing methods that help each one of them or the common problems that they have. It is a bit like business consultancy, where you try to disseminate the best practice across industries. So my background is mathematics. I studied mathematics, and my specialty areas are probability theory and differential equations. And now let me show you the two projects that I work on during this period that is funded by the AXA Research Fund. So one of them concerns model risk. Model risk is a notion that is probably known to some of you from financial mathematics. It is the risk of using the wrong model, even though you deliver the right results from your model. I try to do the same in systems biology. That is, try to find out if, due to missing data or unknown reactants or unknown reactions, your model is not an exact representative of reality, how much your predictions can be wrong. And the second project is trying to estimate contamination in so-called microfluidic devices that one of my colleagues develops. So these are small plates with thousands of little reaction volumes in it on a square centimeter area. He introduces 3 different types of DNA molecules into them and dyes them with the 3 colors, red, green, and blue. But sometimes he notices that there are clusters. Let's say there is a cluster of green colors here, and he wonders whether this is down to contamination, that is one reaction volume contaminating nearby ones. And I developed a mathematical model to be able to estimate this contamination rate. I combined percolation theory, which is a well-developed theory in probability, which means that there are these little white lines. These are connections. These are contamination routes. And I spread randomly red, green, and blue spots on the plate, and then see how, when you have a green spot somewhere across such a contamination route, then it contaminates its neighbors. And from the second image, I try to estimate how much contamination happens in the system based on the first picture. And I'm driven to help biologists help problems that otherwise they couldn't solve. They couldn't solve without mathematical modeling. Thank you.